Good day, my students. We are also here for yet another of our lecture series on staining microbiological techniques, MCB222, to be taken by Comrade Isaac Igbawua of the Department of Microbiology. In today's lecture, we'll be considering dyes and stains used in microbiological staining. This is our week 3 lecture in the series, and in today's lecture we'll be considering why do we have to stain bacteria. We'll also look at what are dyes and or stains. How do you classify, or what are the classification of stains? What is the mechanism of staining? So that at the end of it all, we should be able to know what is staining. Now, why must you stain bacteria? We should also to be able to explain at the end of the lecture what is a stain. What type of stains do we have? How does stains work? So that it will not just be about impacting color to a specimen on a slide and viewing under the microscope that we discussed in our last lecture. Why do you do it? That is what we'll be considering in our today's lecture. And so I want to encourage all of us to be attentive so that you will get what is needed. Dyes and stains, as earlier said, used in microbiological staining. Why do we stain? Most types of cells do not have enough natural pigment and are therefore difficult to be seen under the light microscope unless and until they are stained. Another reason for why we stain is that there are specific staining techniques which are used to determine the cell's biochemical or structural properties. In other words, if you apply some stains, there are some structural or biochemical details that you will not be able to see unless you apply certain specific staining techniques. Some bacteria, as you are aware, have capsules. Normal staining will not bring out the capsules. Structures such as flagella in some of bacteria cells that we know will not be visualized except special staining techniques are employed. And so we stain one to add pigment to see things that ordinarily the pigment is faint and cannot be so much visualized. So we increase the pigment by staining, use of stains. Staining helps scientists to identify and classify microorganisms. When you stain, it will tell you whether it is gram-positive or negative. The stain is a differential type and things like that. So you identify whether it's a cocci or a rod. You identify and classify depending on what you see and depending on the type of stain. And staining also can be used by healthcare providers to diagnose the cause of bacterial infection. As said earlier, tissues can be examined when stained, and the causative agent or organism that is the problem can be visualized following staining when examined under a microscope. A stain, otherwise called a dye, because the term is used interchangeably, is a synthetic chemical which is derived from nitrobenzene or an aniline. Stains are used commonly in microbiology to increase the contrast between microorganisms or parts of its component structures and then the background, so that it can be easily visible. In other words, you use stain to increase contrast between microorganisms as number one, or the individual component parts or structures and then the background. If you have a bacterium lying in a place, the outline, the cell wall will be visualized if stained, because on a general note you will just see something suspended in a fluid. But if you now stain, it brings out the contour and the shape of the bacterium, so that you can be able to actually get the contrast courtesy of the stain. The process of giving color to particular organisms or component of its structures is what is generally known as staining, as earlier said. And each stain or dye is composed of three components. One, as said earlier, is the benzene ring. Number two is a chromophore. And number three is an axochrome. I repeat, stains or dyes are composed of three components: a benzene ring, a chromophore, and an axochrome. The benzene ring is the colorless part of the stain or the dye, and it is the basic structural component of the dye. The chromophore is the functional group of the dye that gives color to the stain. Whichever color that is there, chromophore, as the name is what gives the color, whether purple or blue or green or red. It is that chromophore component of the stain that distinguishes it. Benzene ring and chromophore are collectively known as chromogen. The axochrome, which is the last of the three components in the group, gives the ionic property to the stain. Whether it is a basic or it is acidic stain has to do with the axochrome. Stains are classified based on the nature of chromogen. I've told you earlier that benzene ring and the chromophore collectively called chromogen, and that function is used in classifying stains into three: acidic stain, which is anionic stain; the basic stain, otherwise the cationic stain; and then the one that is neutral is neither acidic nor basic. It is neutral. Now we'll consider acidic stains. The chromogen of an acidic stain is negatively charged, so it is known as an anionic stain. An acidic stain used to stain the positively charged component, such as the background of a staining. Background because the nucleic acid are acidic in nature, and because an acidic stain is acidic in itself, the like poles will repair. So the acidic stain will rather stain the background, which is believed to be alkaline in nature. Acidic stain cannot stain bacterial cell due to repulsion of same charge. I said earlier, you know, a bacterial cell has teichoic acid, so the like pores will not be able to allow acidic stain bacterial cell. Examples of acidic stains include eosin, nigrosin, Indian ink, and the rest. Now we'll consider basic stains, the cationic stains. Chromogen of or colored part of a basic stain is positively charged, unlike the acidic, which we say is negatively charged. And so in this case, it cannot, it is called a cationic stain and cannot be able to stain basic structures, just as in the acidic stain. If it is basic, it will stain acidic. If it is acidic, it will stain basic, and vice versa. So basic stains are used to stain negatively charged components such as the bacterial cell. Examples of basic stains include methylene blue, safranin, malachite green, basic fuchsin, crystal violet, so on and so forth. The neutral stain. In neutral stain, both cationic and the anionic components are colored such that net charge is neutral. Neutral stains are actually a sort of acidic or basic stain. There are sorts of acidic or basic stain. For example, Giemsa stain, the Romanowski stains, as an example. Mechanism of staining. The process of staining involves an ion exchange reaction between the stain and component to be stained. There is ion exchange between the two, the stain that is coming and then the component that has to be stained. There will be exchange of ions for a reaction to be observed. For example, bacterial cell is negatively charged due to large number of protein having the COO negative group. This negative charge is balanced by positive charged ion present outside the cell wall. Therefore, a bacterial cell is represented by, as put in the bracket, bacterial cell, which is negative, and then the sodium, which is a positively charged ion. To obtain the bacteria, to stain the bacterial cell rather, cationic dye are used having positively charged chromogen. For example, methylene blue, which is represented as MB+, which is the methylene blue. So it will now have to attract the negatively charged chloride ion from the background. During staining, bacterial cell is flooded with methylene blue, and due to ion exchange mechanism, acidic component of bacteria, that is the bacterial cell wall, become stained. The reaction occurs as follows: bacterial cell negatively charged positive ion of sodium plus the methylene blue, which is from the dye now, with the supposed chloride ion, we now give sodium chloride plus bacterial cell, which is the negative plus MB positive ion. The MB is the methylene blue. Now, classification of stains. Stains could be classified into either simple, differential, and special stains. Most, if not all, stains falls into any of these ones. We'll consider them briefly, one after the other. Simple stains. It involves the use of only one stain, and all types of bacteria appear as the color of that stain when viewed under the microscope. Some stains commonly used for simple staining include crystal violet, safranin, and methylene blue. The bacteria stained with crystal violet will appear violet color, safranin red, methylene blue, as the case may be. Just use of one stain, that is the key thing there. Use of one stain to impact color to a cell under view, and the cell is made to take the color of the stain. Simple stains can be used to determine a bacteria's species morphology and arrangement, whether it is round or rod or oval, or the arrangement is in chains, or is in clusters, or is tetrad, or whatever. All simple stains, when used, could be able to place the organism so that the morphology and the arrangement could be seen, but they do not give any additional information. Living tissues or living bacterial cells, for instance, are almost colorless and do not present sufficient contrast with the water in which they are suspended to be clearly visible. As said earlier, if not for the stain which impact a contrast, you may not be able to distinguish to say, oh, there is a bacteria cell lying here. There is a bacterium in this suspension, because the differentiation will not be there. But with use of stains, even if it's just one stain, at least it can give the contour, it can give the morphology, and as to whether it is a rod or a cocci or whatever, that can be seen. But internal details and any additional information may not be seen. Differential stains as the second of our classification, having treated the simple stain. Differential stains use more than one stain, and says we have to give a different appearance based on the chemical or structural properties of the cells in question, vis-a-vis the basic components of the stain, whether acidic or basic. Some examples of differential stains are the gram stain, the acid fast stain, and the endospore stain. These are examples. So depending on what stain you are using, the appearance of the organism under examination or will be different depending on the stain. Special stains as the last of what we'll consider under classification of stains. There is a variety of staining procedures which is used in identifying specific external or internal structures that are not found in all bacterial species, such as the capsule stain and the flagellar stain. As said earlier, it's not all bacteria that are encapsulated. And so when you want to demonstrate whether or not a bacterium has capsule, special stains are needed. More so, some bacteria has flagella, whether polar or scattered all over, aspergillus or whatever. The flagella has to be stained to be able to be visualized, and special stains are employed to be able to view the flagella and also classify the bacteria as such. The simple stains, as we discussed earlier, commonly used dyes for simple stains include the crystal violet, the methylene blue, the safranie. And these simple stains use one dye, as said earlier. They are used to provide color to otherwise transparent bacteria cells. At this point, we will adjourn for the class, and you will understand that bacteria cells ordinarily wouldn't have been able to be viewed if not for an increment in their contrasts, because ordinarily they are suspended in fluids in the environment they are found, and they cannot, and we cannot be able to visualize them until a contrast is created courtesy the use of stains. So whether or not the structures will be stained depends on the type of organism, the type of organelle, be it capsule or flagella, or generally the bacterium, and as to whether or not it is the cell wall, which is basic, or the nuclear structures, which are acidic in this case, and the opposite of the stain component will be used in impacting color to such a structure. Thank you for listening as we wind up also for today for yet another lecture by His grace next week. Thank you so much for listening.